Hello everyone, welcome to our Bioinformatics Club's workshop on Python basics with very fun examples. Whether you're new to coding or just new to applying it in biology, this session is designed to give you a foundational understanding of Python using concepts you're already familiar with like DNA, genes, and proteins. So by the end of this workshop, you'll be able to write small, powerful scripts that can analyze genetic data. So get ready to turn abstract code concepts into concrete biological tools. Some bioinformatic basics to know beforehand are strings, which is text, and it's used to hold whole DNA or RNA sequences. The next thing to know would be lists, which are ordered groups, and these are used for a list of 3-letter genetic codes or codons or a chain of amino acids. Dictionaries are lookup tables, so it's quite literally like a dictionary. There is a key and there's a value. It's used to create the codon table, which maps a specific codon like, for example, AUG to its corresponding amino acid like methionine. And the next thing to know would be numbers, which can be of two types, integers or floats. These are used for counting bases or calculating a percentage like the guanine or cytosine content of a sequence. The next thing to know would be for loops, which is repetition, and it's used to look at every single base in a DNA sequence one by one. And there's another concept called if statements, which is decision-making statements. It's used to check for a specific pattern, like asking, if I see the ATG sequence, then I've found a start signal. Usually for and if statements are used together to solve bioinformatics problems. So let's start with the first one, which is if statements. The code snippet performs 3 separate checks, one for each base variable assignment, A, T, and G. Initialization: in each check, a variable named base is assigned a single letter DNA nucleotide. For example, base is equal to A. And then you check for purines. The first condition, which is if base equal to A or base equal to G, this checks if the base is either adenine or guanine. And if it is true, the code prints purine. Purines are characterized by having a 2-ring chemical structure. Next, we would be checking for pyrimidines. If the first condition is false, the code checks the second condition, which is defined by the elif base equal to C or base equal to T, to see if the base is either cytosine or thymine. If this holds true, the code prints pyrimidine. Pyrimidines are characterized by having a single ring chemical structure. Invalid base checks: if neither of the above conditions are met, meaning the base is not A, G, C, or T, the else block executes. This prints invalid base, indicating the input is not a recognized DNA nucleotide. The program's output based on the 3 sequential blocks of code would be checking first base, which is A, which results in a purine, checking the second base, which is T, which is a pyrimidine, and checking the third base, which is G, which results in a purine. Next, let's look at for loops. We are using the same DNA sequence as before. Firstly, we will make a dictionary. Dictionaries are useful for storing large amounts of data and efficient retrieval of values based on their keys. They are in curly brackets, and that's their syntax. They come in key value pairs, which means each item consists of a key and its corresponding value. For example, it's like ATG codon will be mapped to M, which is methionine. So this maps codons to the corresponding amino acid. And the asterisk there represents a stop codon in our dictionary. Before the for loop, we must declare a variable with an empty string to store the protein when it's formed from our for loop. We will use protein equal to quotation marks. For loops, they can repeatedly execute a block of code for each item in the sequence. For example, here for I in range 0, length of DNA sequence -2, 3, which means it mentions the range, which is start, stop, and step, which means how much I increases by. The -2 prevents slicing beyond the end of the string. So basically, this processes the DNA in chunks of 3. For codon equal to DNA sequence, I is to I plus 3. This is string slicing, which means string start to end. This is taking a substring of the DNA sequence string. For example, I equals to 0, which corresponds to the codon, will be equal to ATG. I equals 3 will correspond to the codon CGT. This step is how we read DNA sequence in the same way ribosomes do in real cells. For amino acid equals codon table dot get codon, X, this gets or looks up the codon from the dictionary we made in the beginning. If the codon isn't found, it will add an X for unknown. In our case, all of the amino acids in our DNA sequence were added in the dictionary, so we won't see an X in the output. In the next step, with protein plus equals amino acid, this will add the amino acid to the growing protein string that we declared in the beginning. Now we're all set to print our results. This code is basically a simplified way of how our cells also read DNA sequences. One difference is our cells have to transcribe our DNA to mRNA so it can send the message to ribosomes. But in coding, we don't have to do that because we have the information of the DNA sequences and we also have their corresponding amino acids, and they will form the protein that you get from these amino acids when they string together. Next, we'll be seeing how to define functions. Examples of functions would be print and input, etc., which you've already seen in our previous examples. But when we need the Python interpreter to perform a specific task for us, there's not always a built-in function for it, so we build a function ourselves. The syntax of a function is to use the keyword def and name it in such a way that it answers the what of your task. And then you write your code which explains the how of your task. Once you write your code, you can ask the Python interpreter to perform the task that you've mentioned and specified in your code. But once you define the function, you have to call it for the Python interpreter to perform the actual task. We'll dive deeper into calling the function as we go through the example. Here we are going to build a function that calculates the GC content of the sequence. So first what you do is you define the function, and the brackets are parameters of whatever you want it to analyze. So GC content of the sequence will be going in the brackets. The sequence upper means that it specifies the Python interpreter to identify uppercases only of the letters. Count is again an inbuilt function that counts the elements in the sequence you have specified, which is G and C here in this example. So basically your interpreter will be counting how many G's and C's are in your sequence. Then you define another function called GC percentage. Length again is also a built-in function that measures the length of your sequence or whatever element you have in there. So if it was a word, it would tell you how long the word is and how many letters make the word. Return is called the output, where the Python interpreter will store the output of the function. So now next we define the sequence that we want to calculate the GC content of. The sequence here is the argument. So when you define a function, whatever goes in that brackets is called parameters, and then when you actually perform the task, whatever you put in those brackets are called arguments. It's the same thing. One is a more general idea, which is parameter, and your arguments are more specific to what you want Python to perform. So we define the sequence that we want to calculate the GC content of, and the sequence here will be the argument, like I just said. You have to pass an argument that corresponds to the parameter, which was sequence in the general function when we defined it. Think of the function which has all the general variables, then we define the actual problem or task that we want the function to work on accordingly. So store the sequence in a variable called sequence. Next, we store the function in a variable called GC. It's not necessary to store it in a variable, but if we want to reuse the GC count of that sequence, we can just call GC again. So here we have called the function. Calling the function just means, or it has a syntax where you type the function out and in the brackets you put your actual value, which are the arguments in there. Here that sequence would be the one that we've been using for all the examples with the for loops and if statements. And then you print out your GC content. Alright, so that was it for today. Thank you so much for joining us. We now hope you'll see how easy it is to start writing your own simple yet powerful bioinformatics scripts. If you have any questions, then feel free to message us on our Instagram, which is UFC_bioinformatics_club. For self-reflection on what you have learned today, a worksheet link is also posted in the description box, and the answer key will be provided once you reach out to us. Goodbye and get ready to code some biology.